Well, I'm very pleased. So we can go directly onto our talk this evening. And our speaker is Dr. Kirsty Kemp, a research fellow at the Zoological Society of London. And she specialises in the study of deep ocean ecology. She did her PhD in zoology in Aberdeen, and this was followed by a postdoctoral work in the Natural History Museum, where she looked at polychaetes. Then she went to the zoo on a Leverhulme postdoctoral fellowship, and she is working there now, as you can see from the information in the advertisements. Dr. Kemp is going to discuss the need for continuous and consistent sampling of the deep sea to understand the mechanics driving community patterns, and ultimately, perhaps belatedly, the scale and extent of our own impact. So it's a great pleasure to invite you, Kirsty, to come and address us. Thank you.
Thank you. I'll just see if, is this working again? Yeah? It's working? Okay, is it working okay? Yeah. Okay, thank you very much. Let's just start this here. Yes, hi. Thank you for the invitation to speak here tonight. It's a really lovely honour to speak here tonight and to talk to you all about the deep sea. But it's also a very timely invitation for me because I have to start by admitting I got a little bit lost in writing my way through this presentation because basically sometimes being a conservation biologist can really take quite an emotional toll, and I'm sure that many of you know this and many of you suspect this, and sometimes you lose your way as to why you're actually doing what you're doing, and an invitation like this can really make you look back and remember it's really cool and that's why you're doing what you're doing. And so it was really nice for me to be able to sit back and think about what I wanted to present to you today. And yeah, so sometimes as a conservation biologist, you feel a little bit like this guy floundering in the ocean. And another timely thing that happened recently was I attended a, what was it called, Conservation Optimism Summit in Oxford a couple of weeks ago. And I mean, it's a sort of a sign of the times that we even need to title our summits Conservation Optimism. But it's a proactive thing. It's a gathering of people who are refusing to be beaten down, and probably more than a handful of people who are feeling a bit down and trying to find a roadmap to climb their way back up. And it was an interesting and encouraging meeting. And basically, it reminded me of well, one of the things that came out of this meeting that kept coming up as a topic of discussion was the power of storytelling. And so I was thinking about this when I was writing this talk for this evening, because I don't want to give a depressing talk, and conservation biology quite often veers wildly into the depressing. And as a self-selected group of people who are interested in and love the natural world, I'm pretty sure you know the score on that front, and you don't need reminding. So I thought instead of getting deep into the depths of fishing and what it's doing to our oceans, which is what I spend my day doing, I was going to talk about some of the stories of how we sample the deep, how we actually access this environment. And as research scientists, we're often encouraged, often forced to incorporate storytelling into our, outreach, it's termed, into our project designs and proposals. And I think everybody values this and recognizes its value, but I hadn't really thought until I was at that Conservation Optimism Summit and until recently about the real value of storytelling to us as humans and how we use it as a communication tool. This is my son. He's telling a story to his cousin. I walked in on this the other day, and he'd taken her picture off the fridge and brought it through to his bedroom because clearly the power of the story was so much that he needed to share it with somebody. It was no good just reading it to himself. And stories of the sea abound. I mean, this is just a selection off my shelf. We have literature and stories of the ocean. People are drawn to the ocean. This particular book here is aimed at 10-year-olds, and I've just finished reading it for the third or fourth time because it's really good if anyone has a 10-year-old or an 11-year-old. But these stories and these literature, fiction, they still vastly outweigh the factual stories of the sea. And the factual stories are just as gripping and just as good. So how do we make that leap for ourselves and for children? Well, let's start by going to the environment that we're considering. This is planet Earth, or to paraphrase Arthur C. Clarke, it's clearly planet Ocean. And let's face it, any extraterrestrials coming down to planet Earth are going to hit water. They're not landing in the Nevada desert to wander around Area 51. They're going to hit ocean with a 70% probability based on the coverage of our planet. They're going to hit and this is the Pacific and the Southern Ocean, and they may be the Atlantic, but wherever they hit, they're going to hit ocean. And when they hit the ocean, they're going down to the depths. I mean, the shelf, there's a pointer on here, isn't it? The shelf and the slope constitute a very small percentage of our oceans. The vast majority is deep, abyssal, and bathyal floor. The average depth of our global oceans is 3,000 meters. 50% of our oceans are below 3,000 meters. And the max depth is approaching 11,000 meters in the deepest, darkest depths of the Mariana Trench. And down here, it's cold. It's dark, and it's heavy. Heavy as in you've got 4 kilometers of ocean pushing down on you. It's approximately 400 bar of pressure. And there's these guys swimming around. This is Coryphonoides aromatus, the rat-tailed grenadier. My first love when I did my PhD, and he's everywhere, the cosmopolitan. Not so keen on the Arctic and the Antarctic, but possibly with the changing oceans is probably even wandering in there every now and then. But yeah, welcome to Earth. This is the classic Earthling that most anybody landing randomly on our planet is highly likely to meet. But his environment, the deep ocean, constitutes an astounding 90% of the habitable volume of our planet. 90%. And we, as humans, as far as we know, the only beings conducting research on this planet and scrambling to publish it and present it at evening meetings, we have a remarkably difficult time getting down to this environment to have a look, this 90%. So how do we get there? How do we have a look? Oh yeah, talking about a remarkably difficult time. And this is Ahmed Gaber. He's an Egyptian diver and explorer, and he currently holds the world record for the deepest dive, the deepest a human has gone. And I realized on the way here tonight that the 5-year anniversary of that record was just yesterday, September 18th, 2014. And he managed to dive to 332 meters, which is mind-boggling. The attempt took him 14 hours. It took 30 people, including 9 other support divers, a mixture of oxygen and helium and nitrogen, and a risk of a multitude of physiological illnesses. I say it took him 14 hours. It actually took him 15 minutes to get down to depth and 13 and a half hours to get back up again. Such are the perils of scuba diving. And he was aiming for 350 meters, and he stopped at 332 because he was feeling the effects of high-pressure nervous syndrome, which is also known as the helium tremors. And this is when you get tremors and visual disturbance and decision-making problems, and somnolence, which are the extreme desire to sleep, which I think is the scariest one. I mean, at 330 meters, you do not want to be going to sleep. So he had already broken the world record. He came back up. And this is our physiological limit as humans. Only 2 people on the planet have ever even come close to this depth. But let's take a look. Here's Gaber's record. And this is not exactly to scale, there's the depths of the oceans. Now, we've managed to map the depth of the seafloor, the best global maps of the seafloor are at a 1 kilometre scale, grid scale. By contrast, we've managed to map the surface of Mars, which is 54.6 million miles away at its closest. On average, it's about 200 million miles away. We've managed to map the surface of Mars at a 10-metre scale, an order of magnitude difference to what we've managed on our own planet. So we've got a ways to go, and obviously as humans we're limited, but our technology is not, and our technology is rapidly expanding and rapidly advancing, both to explore and to understand, and also to extrapolate resources from the sea. And those need to keep up with each other, or the former needs to keep up with the latter. So the primary tool, and the one that I'm going to be talking about most tonight, is clearly the camera. The camera, if used well, can yield data whilst making very little impact, doing very little damage to the seafloor or to the ocean itself, and can facilitate some really good stories. What did I do here? Yes. So it was the camera that introduced me to this field of research as well. I attended a talk when I was looking for my specialty. I attended a talk by Professor Monty Priad at the University of Aberdeen. I think some of you here probably know him. I don't remember what the talk was or why I was there, but I absolutely remember the content because I was hooked, and consequently ended up doing a PhD with him and studying it since then. But he was talking about using cameras to study to answer questions about what was going on the deep sea floor, 4 kilometers underwater. And he was trying to answer questions like, what's there? Where is it? When is it there? How fast does it move and how far does it move? And what's it eating? But then also some things that you might not imagine you could use a camera to study, such as how much energy is it using? What's its maximum power output? These kinds of questions that lab had about animals living so remote from us. So I was hooked on that story, and I wanted to show you some of these stories, because it's old footage from the 90s, and it's dark and it's grainy and it's wobbly, and it gives you I think, a much better feel for what it's actually like down there and what it's like to try and work there, as opposed to some of the really pristine footage that we see on our television screens now. The technology has really come on. So now, what did I need to do, Leanne? I needed to press escape. Yeah? So I just wanted to, and then to the right. Hey, okay. So I just wanted to show you some of the videos. Let's start with this one. What you're looking at here, and I know it's grainy, and it's a bit grainy because it's been converted a few times since it was taken, but it was never great in the first place. But what we're looking at here is, it's a downward-looking camera, and it's looking down on a weighted and a baited cross. And this land, they're called landers, these camera platforms. This one, so it's hovering, if you can imagine, it's hovering above the seafloor like this. This one was deployed in South Georgia, or the Falklands, I think it's South Georgia, this one. And the aim of this study was to try to look at the Patagonian toothfish, which you see swimming in here every now and then. You see a few of them. This is one here. This is a commercial species. It's also known as the Antarctic Cod. And between the 80s and the 90s, the fishery for these things just exploded, and it was fairly unregulated and a lot of illegal and unreported landings. And so there was a real need to try to get a hold on this fishery and manage it properly. And this work was some of the first work that managed to collect data independent of fisheries data, independent of actual catches, on numbers of fish and the size of fish and things that you need to plug into programs to try to calculate stock assessments and things like that. You'll have noticed from that video, you never quite know what you're going to get with cameras. And if I remember rightly, a side effect of this work was interest in a new fishery for the stone crab, because there's loads of them, and that wasn't exactly the plan. A whole new fishery sprung up from that. But incidentally, that fishery, the Patagonian toothfish, is now extremely well managed. It's one of the best well managed that we have. So from these lander platforms, you can get an idea of what fish are coming, what they're doing, roughly how they're behaving, and then you can adapt that to your purpose. And so I don't have a video of it, but the next step was to enhance the bait with transponders, so that these scavengers fish, they eat anything, so they would easily chomp down these transponders, and then with a receiver on the lander platform, we could track where the fish were going within reason, and whether they were coming back to the lander, whether they were coming to feed once and then leaving, excuse me, and get a really good idea of how these fish were behaving and swimming, and what speed they were swimming at, and just general foraging behavior. And then we could adapt it further. This one. So this is the same setup with a bait and a downward-looking camera. But here we have two poles that, I'm not sure if let me play it again. We have two poles that deliver an electric shock between them, a mild electric shock. And you'll see that both those fish, actually one of them does the escape response and it bumps into the other one, which then does an escape response too. So that escape response is called a sea start. The fish, a stimulus stimulates the fish, and the muscles along the opposite side contract. The fish goes into a C shape and shoots off away from the stimulus. And it's the fastest motion that a fish can make. It's almost always involuntary because it's an escape response. But it's the fastest muscle motion that fish can make. So with that footage, we could then digitize that movement, digitize the fish going like this and swimming away, and calculate actually its muscle power output. On an animal that's 4 kilometers under the ocean, this is what I had been doing previously as an undergraduate in the lab with a camera and minnows in the lab. I still think, this was rather a long time ago, and I still think these are amazing, as you can tell. This one was another setup. So we knew when the fish were likely to come to the bait, and a pre-programmed Perspex trap was designed to land over the bait when there were likely to be fish there. So what you see here, again, downward looking, this trap is just landing and sealing the fish inside this Perspex chamber, and that essentially is a respiration chamber. So within it, and you can't quite see where, but within it are instruments measuring the oxygen content of the water that's trapped within that box. So over time, you can measure the reduction in oxygen in that water, put that together with the activity that you see and how many fish you see, and actually calculate the respiration rate of these fish. Again, on the bottom of the ocean. And these are fish that would not survive. We can't have them in the lab. They don't survive the pressure difference coming up from 4 or 2 kilometers onto the surface. Their bodies can't withstand that pressure change unless they're brought up in a pressure chamber. I meant to stop it before that bit. Unless they're brought up in a pressure chamber, which is a hugely expensive and difficult thing to do. Right, I think that's... I usually get a lot of questions on this video afterwards. Yeah. So let's go back to the talk. Which way do I do it? This way. Those are the videos. All right. I've got one more video to show you. But, yeah. So those landers, those were the stories that brought me into the field of deep sea research. And I thought those landers were amazing. But they're still only really snapshots. They're a matter of hours or a matter of days. As I should have said, the landers themselves are released off the side of the ship and they sink down to the seafloor. They're completely autonomous and they operate there for a matter of hours or days before the ship comes back and recovers them. And I wanted to know what was happening over a longer period of time than this. Because let's face it, the Earth has turned on its axis 15 times 10 to the 11th, and I had to sit this morning and work that out. It's 1.5 trillion times. Turned on its axis 1.5 trillion times since the beginning of life on the planet. So life living on this planet has had the signals, the time signals that come from this turning and the rotation around the sun. These time signals have been working on the organisms that live on Earth since throughout their evolution. And these time signals are light and dark, temperature, the temperature changes, and the gravitational pull of the sun and the moon. So time, and I think everybody living on the surface would agree, is fairly critical to our existence. Let's see. I'll come back to the time scales in a minute. But within our bodies, we rely on time signals and synchronicity, cyclic synchronicity, where our hormones are produced in cycles, our respiration occurs in cycles, our heartbeats, our neuron spiking. And on top of that, our sleep-wake patterns occur in cycles, and other whole organism cycles like reproduction, seasonal reproduction, or growing cycles. And these things are really a part of life, and it's sort of obvious to us who live on the surface. At a greater scale, at a population scale, time signals from the environment also entrain whole population biology, like migration patterns, breeding cycles, and predator-prey interactions. But as I showed earlier, the deep sea is dark. I mean, light is completely attenuated in the first 1,000 meters. It's completely dark beyond then, apart from bioluminescent signals. And it's cold. The temperature fluctuations are very, very slight. And we think of these as the two main time cues that entrain our own biological rhythms. So what about these guys? Are they just wandering around? You know, do they have anything in training them? Do they have a day and a night, or a season, or a year? And I really thought that we needed to leave our landers, our observation platforms, down for a lot longer to try to get a handle on what was going on over a longer period of time. Because the sun is not our only signal. The five main signals, environmental signals on Earth, are a 24-hour day, a 12.4-hour tide, a 14.8-day spring neap tide, 29.5-day lunar month, and a 365.25-day year. Those are our five signals that keep us in check. So which of those, if any, were reaching the deep ocean environment and keeping these populations in check? Excuse me. So we built a lander that could stay down for longer. And this was our first long-term lander in all her beauty. She was designed to go down for up to 6 months, to stay down for up to 6 months. Things rust down there, so she had to be completely redesigned from all the other landers in titanium, with all titanium bits, in order to stay down there long enough for us to see what we wanted to see. And right, now I'm going to go back and show you the first video that... Am I doing this wrong again? There we go. Dobo. So this is the first, it's not a video, it's a time lapse of stills photographs that were taken every 3 hours of a porpoise carcass that had been placed on the seafloor. We needed a bigger bait to last a longer period of time to see what was going on. So that was a whole chapter of my PhD right there. And what you saw what you saw was we put the lander down for 6 months, but I've only shown you 10 days because that's when all the action occurred. The carcass was completely gone in 10 days. And there were 3 species of scavenging fish, 1 species of squat lobster and a benthic octopus. That's what you saw. And so as I say, all the action was over in 10 days. That didn't mean I didn't have to still sit and digitize the thousands of other photos just in case. And so we weren't really seeing much longer. Well, a week was still pretty good compared to the couple of days that we'd had before, but we weren't really able to see much. It was a really interesting scavenger study, but not very informative in terms of any other population cycles or anything. This red tinge, by the way, has nothing to do with the deep ocean. This was because, remember I said we were using film cameras, not digital cameras, and we put the film in, I say we, it wasn't me, put the film in the wrong way around, so the emulsion was the wrong way around. We came back 6 months later, picked up the lander and thought that we didn't have any images, but credit to him, the guy who put the film in the wrong way around also then took the film to a series of professionals and managed to extract the images from it. Unfortunately, with this red tinge, which I had to sit in a dark room looking at thousands of them. But so the lander was like a kickstart almost to try and do a lot more observations of long-term signals in the sea. And we looked for many, and I have several stories I want to tell you, but we can't stay here all night. And there's one that I particularly want to tell you because I think that it's the basis of a lot that is to come. It doesn't actually have anything to do with cameras, but it's such a great story that it underpins a lot more of what we need to do that I want to tell you about it. And it was undertaken by Professor Hans-Joachim Wagner, who works at the University of Tübingen. And we were lucky enough to share a few cruises with him at the beginning of my career. And he's an anatomist. He was really interested in whether these deep-sea fish were producing their hormones in a cyclical pattern like we produce ours. Let's see. Oh, yeah. So ignore the cool shark. He was working on these eels. These are also cool fish. This is Synaphobranchus cowpie, the arrowtooth eel. And the reason he was working on these was they survive the pressure change much better when they come up on deck. So they were actually alive when they weren't happy, but they were alive when they came up on deck. And so I looked for some photos of us on deck because I know I have them somewhere. But again, they're printed photos. They're not digital photos. I couldn't find them in the stack. But it was quite funny whenever the fish came up on deck, because we were doing fishing as well. Everybody would just stand back and let Joachim dash in with he had these glasses for dissecting things with 3 layers of lenses. He'd have them all ready on his head, and he'd dash in and grab his fish, put them in a black bag, because he didn't want them to be exposed to a light signal. And light is just an incredibly powerful environmental signal, and he wanted to reduce the possibility of their brains seeing any light, because he was interested in the pineal gland, and the pineal gland sits right on the top of a fish's brain and is easily exposed to light through their thin skulls. So he'd grab these fish, he'd put them in a dark bag, he'd take them into the lab, and then he would dissect out the pineal gland and put it in a Ringer solution. And the Ringer solution is designed just to keep the gland happy, make it feel like it's still in the brain and comfortable. And he would leave the gland there in its Ringer solution in the dark, and he would come back every few hours and change the Ringer solution and measure the melatonin content in the solution that he'd taken out. I forgot to talk about, wrong way. Forgot to talk about melatonin. Melatonin is a hormone which we have as well, which regulates our sleep-wake cycle. For us, we produce it at night, and it degrades during the day, and it is strongly linked with light for us. And Jochen was going in and measuring the melatonin in these solutions over time. And he actually managed to measure changing levels of melatonin being produced by these glands over the period of hours that they survived. And I was part of this because I happened to be using the lander I showed you to measure ocean tidal signals in the same area of deep sea that he was collecting these fish from. So I had my lander down there measuring ocean velocity and current and finding a very, very strong 12.4-hour tidal cycle at the bottom of the sea, right where he was taking these fish from. So we wanted to put the data together. And the first thing he did was look at the melatonin cycle, peak-trough cycle, in the context of a solar rhythm, because that's what we know. And it didn't seem to show any rhythmicity at all. So he then looked at it in the context of a tidal rhythm, and it was perfectly synced. And I just think this is amazing. First of all, that he managed to do this, but second of all, that it's synced to a tidal rhythm, because a tidal rhythm, I mean, a few steps away, maybe it has something to do with the sun, but it's not linked to our light-dark cycle, which is what we thought melatonin was directly linked to. So this just tells us there's so much going. I mean, it certainly doesn't say that they're absolutely entrained by a tidal cycle, but the two are very much in sync. And it's the strongest signal that I was finding, physical signal anyway, that I was finding in this area of the ocean. And it just tells us that there's so much more about this physical environment that these fish could possibly be reacting to, that their own biological clocks could be entrained by, that we really don't understand. So is that really important? I mean, is it important whether they can tell time or not? This is maybe a tenuous link, but I think that there is a link here. If you think about animals responding to external signals, exogenous signals from the environment, it tells us something about how they respond to pressure. And you have to bear with me with this slide. I know it's terrible, but I tried to make it a little bit more interesting. It's actually quite difficult to draw in PowerPoint, so that's supposed to be a straight line. If you think about a state, let's say a population at this level, and an external force is applied, the population might peak and trough and wobble back and forth until it reaches its natural equilibrium again. Or it might peak and show a decay response. And these are some of the responses we see to external signals like those I've been talking about. But these are also some of the signals we see in response to external pressures like fishing, for example. Because they don't always respond like this and regain their natural equilibrium. Often they can be knocked out of whack and regain and establish an equilibrium at a different point. You know, this is where the natural population was, and some force has been applied, and the population has now shifted to a different state. Now, these very large continuous environmental forcings, a very good example of one of those is fishing. And it's often a pressure that populations just simply can't withstand. Whoops, that's not what I wanted to do. Oh yeah. Just simply can't withstand. Once fishing pressure has been applied, we often see a population regaining stability, but at not at the point it began. Escalating climate change is another example of this type of impact, and both of these things are hot topics in the Arctic right now, which is where my current research is. So, some years ago, my research began to swing from this basic research that I've been telling you about, basic as in studies that are aimed at expanding the base of scientific knowledge, towards applied research, or research that's designed to solve a specific or practical problem or answer a specific question. And based on the fact that we'd been doing some of this camera work in deep water, I was approached by the overarching body for Greenland's fishing. It's a management body called Sustainable Fishing Greenland. Excuse me. And they approached us to ask whether we could help them, whether we could design a project, a scientific approach to trying to figure out what impact their industry was having on the environment in which they worked. And most of the fishing off West Greenland is trawl fishing. This is an example of a trawl net. They are traditionally the most destructive of fishing gears, and they drag along the seafloor behind the boat. And we often in our footage see these trawl marks which are left on the seafloor, excuse me, behind the trawl nets. In fact, you can see there's satellite images of the North Sea which just show these trawl marks crisscrossing the entire North Sea. So we can see these from space. So it's a destructive industry, and many people, I often get asked, because I usually give a talk that's much more focused on fishing itself, and I often get asked, why can't we just ban trawling? And there are lots of movements towards banning trawling in certain depths or banning trawling in certain areas, and maybe ideally one day we will be able to do that, but first we need to recognize that this is a hugely important industry to Greenland. It constitutes or underpins 25% of the employment in the country, including fish processing, not just fishing, but also fish processing and landing it and shipping it and making other fish products. 90% of their total export revenue comes from fish and fish products. And shrimp, which we were working specifically with the shrimp fishing industry, constitutes 60% of this. So this is the country's total export revenue. 60% is based on shrimp. They call it red gold in Greenland, these little red shrimps. And you might be thinking, well, why is a science team in the UK doing this work? And partially it was because we had some background with the camera work, but also there's a really interesting connection between the UK and Greenland when it comes to fishing, because the UK is Greenland's biggest market. We import 50% of global cold water shrimp, and most cold water shrimp are coming from Greenland. Royal Greenland is the UK's biggest seafood supplier out of everything that we eat, and it sells about 600 tons of cold water prawns to the UK market annually. And this is important because the UK market, when you compare it to consumers globally, we here are significantly more concerned with the chain of supply and the sustainability of an industry. Our top 4 supermarkets are all competing with to wave their sustainability flags and put sustainability badges on their products, and that's driven by our choice. And although you know, I said at the beginning, you can get a bit sort of bogged down thinking about this stuff. It's a really positive story to think that actually in Britain we're doing quite well. Driving our supermarkets, who are then driving the fishing industry, because the fishing is an expensive process to enter into a sustainability certification, and it's an optional one. And they were doing it because they risked losing their UK market base. They were also doing it, I found out over the course of 10 years, because they really cared. That's not what I would thought at the beginning, I have to admit. I went in skeptical, and I came out really happy with our collaboration with this industry, because they really genuinely do care about the impact that they're having. So we took the challenge and we went to Greenland, and we designed a camera. It's a little bit different. This one's not autonomous. It remains connected to the ship on a winch wire. But it's very similar. It's a downward-looking camera here, and a flash and a battery unit. The trigger is this little weight in the corner. When it hits the seafloor, it triggers the camera to take a picture, and we sail around, putting the camera up and down, taking pictures of the seafloor and waiting a few minutes in between to let the silt settle. And we went out and we did a test study to see if this would work, whether it would yield anything useful. And it did. So we took the opportunity to sail further up the coast and to choose our spots. So this is us choosing a spot to deploy the camera. That's the camera being deployed. And then it comes back up on deck. We can't see anything while it's working. It comes up. We take the images off for analysis. And it worked so well. I'll show you some of the images. I'll get to that in a minute. This is a picture of the power of the camera in action, because these fishermen, they've been working in the region their whole lives. And I was on their vessel taking pictures of the seafloor, and nobody's ever seen pictures of this part of the seafloor because nobody's ever taken them before. So I'd taken the lander up, and I was in the cabin, the side cabin, looking at the pictures, and suddenly I had 5 people looking over my shoulder. And it was so great. And these guys were supposed to be shooting the next trawl, which is a really dangerous activity, and everybody's supposed to be in position on deck when the alarm goes for the net to go out. And they were in totally the wrong part of the ship, looking over my shoulder. And I love it, because that was all of us seeing the seafloor for the first time. And like I say, these guys have been working their whole lives there, and they've only ever seen the mangled animal on the deck of the ship when it comes up, and they don't look the same. They don't look like something really worth caring about when they come up on the back of the deck. So this was hugely rewarding for me. These are some of the photographs that we were taking, and Greenland's a big place. We've been trying to take these photographs. It's essentially like throwing a quadrat, in basic biology, out in a field of daisies, throwing your quadrat. But we had 2,000 kilometers of coast to try and throw our quadrat over, and it's taken us a few years, but we've got really good coverage of the West Greenland coast now. And we see these different communities. We see, what have we got here? We've got this first community, we've got sea squirts and sea cucumbers. Here we've got sea anemones and ribbon worms, snails and shrimps. These were the target organisms for the fishery, polychaete worms and starfish and various things. But you can also see from this footage that we get a really good idea of the substrate as well, not just the communities that are on the seafloor, but also the substrate. Is it muddy? Is it rocky? Is it steep or flat? And so we set about, with thousands of images. And essentially the basics of what we're doing is looking in these images, looking for life, and then labeling it. So here's our image, and here's all the life that's in that image. Also polychaetes in that one. And I don't actually know how many images we've taken now, but we've covered, my colleague's not going to like me for this, but I'm going to say 60 plus stations along the 2,000 kilometers of West Greenland. And we've been taking these community assemblages that we've been establishing, and we've been putting that data together with data that were provided by the fishing industry. So what you see here in the first map, these orange marks, if we were to zoom in, these are made up of tiny little lines, and each line is a trawl that's been undertaken at some point during the past 40 years. This map was a GIF. It's not imported as a GIF, so I can't show you, but if it was to play between 2000 and 2011, I think, you actually see these orange marks moving further and further north, and that shows us that the intensity of fishing is moving north, and they're following the shrimp, and it's not clear to us or the fishermen whether the shrimp are moving northward because they've been fished a bit too heavily in the south and they're just doing better in the north, or whether they're moving northward as a result of changes in climate. But whichever way, they're moving northward and the fishery is following them. And we're scrambling to get to the very north before the fishery does, so that we can see what it looks like before it's impacted. But the fishery have provided, this map only starts at the year 2000, but the fishery have actually provided 40 years of their trawl data, which is a really valuable data set to know exactly where they've been, and also to know which areas have been trawled in the past and then have been left alone for 5 or 10 or 15 years, and we can try and see if there's any signals of recovery there. And on the right is a map of substrate types that my colleagues have been producing. So from the images, we've also been extracting information on substrate type and making a habitat map, a habitat suitability map, to put these three pieces of information together to try to understand what impact has already been made over 40 years of fishing, what impact or what recovery might be possible, and more importantly, perhaps, because this region has already been heavily fished, but perhaps more importantly is what can we do in this northern region to kind of freeze the footprint of the fishery where it is, so that we can try to avoid damage to the ecosystems of the seafloor before the fishery reaches those regions. And we've had some major successes. The Greenlandic government has closed an area in the north to fishing on the basis of our scientific advice, which is a real win. And the trawl gear has been adapted somewhat. It's constantly being adapted. But over the course of this project, they've changed the gear slightly so that the chain I showed you, which rolls along the seafloor, that's been lifted a bit higher up off the seafloor, and they call them tickler chains that hang down from that to still reach the seafloor to encourage the fish to swim up or the shrimp to swim up into the net. So they've added these tickler chains, I love that name, to do theoretically less damage than the original chain which drags along the seafloor. And it does do less damage. It's a step in the right direction. And I mean, I'm happy to talk more about this project, but I forfeited some of the details so that I could show you some of the stories that brought us here. But it's current work, and you can find it if you just Google ZSL Greenland, you'll find it. But I also wanted to show you another story that came out of it, because as part of our project design, in order to get the money from the EU that we were doing this with, we said we would develop a game. We've been going into classrooms in Greenland and classrooms here in London, or in the UK, to teach children or engage children in stories about the seafloor. And so one of the ways we decided to do this was to design this game called Tricky Trolling, where you can be a fisherman, and your aim is to fish sustainably and not damage the seafloor. And the game has been developed in Greenlandic and Danish and English, and it's been really successful. Kids love it, but adults love it too. I took it to that Conservation Optimism Summit a few weeks ago, and one woman played it for 2 hours straight, and by the time we were closing up our stall, I heard her telling our blurb about the Greenland project to somebody else, and I heard her say, I don't actually work on this project, but I've been standing here so long I feel like I can tell you what they've been saying. She was playing this game for 2 hours. It's been a really powerful tool. And before I wrap up, I'm not quite finished, but I wanted to introduce some of the people who are doing this work. And my primary colleague at ZSL is Dr. Chris Jessen. He's been running this project with me in Greenland for several years. Soon to be Dr. Stephen Long, he's our PhD student, and Dr. Mona Furman. And this is us at a trade fair in Greenland last year. And we've been working together with the Greenland Institute of Natural Resources in Nuuk, which is the biggest research institute for natural science in Greenland. And I always like to mention the officers and crew of the research vessel, the research trawler Pamiet, our beautiful trawler that we've been working on for the past few years and was scrapped last year. It didn't pass its MOT, so it is no longer 60-year-old trawler, and I loved it. But it is no longer. And Sustainable Fisheries Greenland, who started the whole project really, and irritatingly for the purpose of talks don't have a logo, so I just had to put, I've been trying for 6 years to get them to develop one, because you need one in talks. But, and I wanted to mention Ocean Lab in the University of Aberdeen, because those were the first video footage that I showed you, and those landers, they were all developed at Ocean Lab at the University of Aberdeen in Montypriad's lab, and they were the beginning of everything for me. So all of these people are devoting their lives to working in, to accessing and working in and understanding the deep sea. And it's a place that remains remote to so many people, and it always will be so in reality for 4 kilometers of ocean. But it doesn't have to remain so in people's minds and in our knowledge, and in what people understand and care about. This is a picture again of my toddler. This is his underwater camera. It was mine, it's now his underwater camera. And this was the first of many times he carefully buried it under some seaweed or some rocks that he found interesting, and to go back searching for it. And it just reminds me that we have the tools. We have the tools to see our planet properly. And really we know enough to care. We've got no more excuses. We know enough to care. And we know more every day. And so I just wanted to close with that, with this classic quote that conservation biologists will almost always close a talk with. In the end, we'll conserve only what we love, and we'll love only what we understand, and we'll understand only what we're taught. And that was what I was thinking of when I made this talk tonight. I just wanted to show some of the stories that made me love this work. And I know you all do already, or you wouldn't be here, but hopefully some of it's engaged you as well. Thank you very much.